Hello Professor Karen and my dear classmates. My name is Harpreet Kaur Virk and today is August 9, 2019 and this is the week 6 of microbiology class, that is Bio 225, and the title of the experiment is Environmental Influences on Microbial Growth and Salt Tolerance Testing. And this is actually the second video related to this experiment. And in the first part I inoculated the 6 tubes and I used the 2 microbes and 3 tubes for each microbe with different concentration of salt, that is sodium chloride. So there were 3 tubes for each microbe with 1% sodium chloride, 7% and 15% sodium chloride. So I inoculated them and kept at them for 48 hours and then now observed the microbial growth in the tubes. And this experiment actually helped me learn like the microorganisms, they prefer more salt concentration environment or more without salt. So what I observed was that in 1% salt solution for both yeast, that is baker's yeast, and S. epidermidis, I see the 1% one here, and this is the 1% sodium chloride. So you can see in the base there is a little bit of slight microbial growth and very slight turbidity. So that shows me that 1% NaCl is preferable for both the microbes. And then I saw in 7%, so in my tubes they seem to be clear and maybe very slight, not as compared to the first tubes. If we see from far away they almost look clear, and if we are close I see very slight in the S. epidermidis but not in yeast. So this means that as we are increasing the salt concentration, the growth of the microbes is not that good, it's restricted. And when I go to 15% for both of them, they are almost the same as I started in the beginning. They are same as clear, not a single, flake of the growth of microbes. So this experiment actually helped me learn, understand how the microbes, they are very specific about the environment, and how these 2 microbes, they are not able to survive in high concentration of salt. That means they are halotolerant, we can say, because they don't need salt for, growth. So they prefer more the environment which is less salt. Like S. epidermidis, it doesn't prefer more salt, and I can understand it's on the skin, it's a bacteria of the skin and in the armpits, and then because our skin has a little bit of sodium, by sweat, where sweaty areas are, so that shows 1% is favorable for its growth. And that was a very interesting experiment, and it helped me learn a lot about what environments they favor the growth of these microorganisms. And I hope we are going to do more experiments like this in the future weeks. And that's it. Thank you very much.